Hello everyone, and welcome to the non-coding RNA webinar titled The Competitive Endogenous RNA Network in Cancers. My name is Daniel Yosub, and I'm a journal relations specialist for a non-coding RNA journal. Please note that this webinar is being recorded and will be made available as soon as possible. Today we have with us a great speaker who will share some fascinating research on this topic. Before we get started, I would like to provide a brief description of who we are. MDPI has been a pioneer in scholarly open access publishing since 1996. Our headquarters is in Basel, Switzerland, and our mission is to foster open scientific exchange in all forms and disciplines. A few words about our journal. Non-coding RNA journal is an MDPI journal dedicated to the publishing and global dissemination of peer-reviewed research in all areas related to the function and the biology of regularly non-coding RNAs. Currently, our impact factor is 3, the site score is 7.4, and the time to first decision is around 23 days. Regarding our program, here is a short overview of our webinar. For today, we have Professor Michael Ladomeri as our chair and Dr. Davide Barbagallo as invited speaker. We will have a Q&A session at the end, and we encourage you to let us know your questions. I also want to mention that any time during the webinar, the audience can type question in the Q&A chat, and our chair will go over each question during the Q&A session. Thank you. Now I would like to invite our chair to introduce and just have a few words. Thank you.
Hello, everyone. It's lovely to have you here for this webinar. It's my privilege and pleasure to chair it today. So my name is Michael Ladomeri, and I'm a professor of genetics with a special interest in RNA biology. I work at the University of the West of England in Bristol in the United Kingdom, although I'm originally Italian-Australian, just to make things more interesting. So just a couple of very quick notes of personal reflection. I will then introduce Davide and pass over to him. So I entered the RNA world in 1991, studying a PhD on maternal messages. And in those days, the RNA field was a kind of fringe pursuit of a few fanatics. And also in those days, we had no idea, literally no idea about all these amazing regulatory non-coding RNA networks. We literally were completely naive to it. And winding the clock, 34 years forwards, it's just incredible to see where we are now. And, Davide's talk will, underline the amazing complexity of these regulatory non-coding RNA networks and their involvement in disease. So enough about me. I will now just spend a few minutes introducing Davide. So thank you very much, Davide, for giving your talk today. We're very much looking forward to it. So Davide Barbagallo is an associate professor of experimental biology at the University of Catania in the lovely island of Sicily in Italy. Since his PhD, Davide has been interested in the study of dark matter of the genome, and more specifically in non-coding RNAs and their involvement in cancer. His first study focused on microRNAs, and since 2015, after the first reports of the discovery of circular RNAs, he helped to consolidate their involvement in glioblastoma pathogenesis as well as other diseases, mainly pointing at competitive endogenous or ceRNA networks. Thanks to a fruitful collaboration with the group of Jørgen Kjems and Thomas Hansen, during his postdoctoral fellowship sponsored by the Umberto Veronesi Foundation at Aarhus University in Denmark, in 2017, Davide cloned the sequence of SMA-RCA5 circular RNA into an expression vector and showed the tumor suppressive function and mechanism of action of this circular RNA in glioblastoma multiforme, one of the most malignant brain cancers. This research allowed Davide and colleagues to be awarded with the Best Paper Award in 2018 from the International Journal of Molecular Sciences. In 2018, Davide also went back to the University of Catania at the Department of Biomedical and Biotechnological Sciences, where he was able to continue his passion for circular RNAs. Davide is the author of more than 60 peer-reviewed scientific articles. So in the present talk, which we are about to enjoy, Davide will show unpublished data on a relevant feed-forward gene expression loop involving a ceRNA network, potentially responsible for the relapse of meningioma, another common brain tumor. Please enjoy the webinar. There will be plenty of time for questions at the end, but if you don't mind writing them in the Q&A window rather than asking them by voice. Thank you very much, and Davide, over to you.
Thank you very much, Mike and Daniel, for your presentation. It is a pleasure and an honor to speak today about these data that, as Mike said, ceRNA network involved in meningioma recurrence. So, first of all, the first question that all the audience and first of all, myself, ask is what is a ceRNA network? For the first time, ceRNA network has been used as term by the research group of Pandolfi when they published an essay on the so-called Rosetta Stone as a hidden RNA language. As Mike said during his introduction, the RNA biology is really an exploding field in the last years, and all comes from so-called dark matter of the genome. Dark matter of the genome, that is the part of the genome that firstly were not known about their function, and only in the last years we are becoming to better understand how this piece of the genome works and how the RNA that are synthesized by this piece of the genome exert their function. Briefly, the ceRNA network can be depicted through this slide. As you can see on the left of the screen, there is the classical post-transcriptional regulation performed by microRNAs, as we'll see later. That is, the messenger RNA is transcribed into the nucleus, it crosses the nuclear membrane, and then in the cytoplasm it can be regulated by microRNAs and the so-called RISC complex that together with microRNAs can either degrade the messenger RNA or otherwise can block their translation. But what happens, and now you can see the right part of your screen, if a third type of non-coding RNA molecule that you can see as just I show the pointer, okay, that you can see as these molecules of RNAs with red boxes inside. So if a third type of non-coding RNAs enters into the cell network. If these red boxes represent binding sites for microRNAs, microRNAs can be attracted by these sites exactly in the same way they are normally they normally bind their natural target, the natural mRNA target. So in this manner, these third molecules can indirectly activate the messenger RNA through the action of sponging microRNAs. And to better understand these networks, we have also to speak briefly about the actors involved in this network. And more specifically, the first actor, as mentioned before, is represented by microRNA. MicroRNA are quite at the top of the regulatory gene expression regulatory network in the cells, and two Nobel Prizes in 2006 and 2024 related to the field of microRNAs. In 2024, the Nobel Prizes, Victor Ambros and Gary Ruvkun, have been awarded by Nobel Prize for the discovery of microRNAs. In 2006, Andrew Fire and Craig Mello were awarded for the discovery of RNA interference, that is the mechanism through which microRNAs work into the cell. But how they work, as I told before, microRNAs, after a complex pathway, biogenetic pathway, exert mainly their activity into the cytoplasm of the cell by direct interaction with mRNA molecules, and especially at the 3' UTR untranslated region of microRNAs, where they can mediate messenger RNA degradation or translational inhibition. The second actor of the ceRNA network is made by these other non-coding RNA molecules that can be of various types. More specifically, in this webinar, I will focus on circular RNA. You can imagine that quite all the other non-coding RNAs, like long non-coding RNAs, pseudogenes, and other non-coding RNAs potentially can act as I will say about circular RNAs, but because of the main focus of the webinar, I will speak about these molecules, circular RNAs. Circular RNAs have been discovered for the first time as a class in 2013 by two different groups, the group of Horgen Kielms in Denmark and the group of Rajewski in Germany. And since then, lots, more than 80,000 of circular RNAs are encountered in the databases and are stored in the database as a sequence. They can act in several ways, through several ways into the cell. Mainly, they have been discovered as sponges for microRNAs, so they exert exactly the same function that I mentioned at the beginning of the presentation, but also we ourselves discovered the action of one circular RNA as a sponge for RNA binding protein, for a splicing factor most specifically, and also they can act as a scaffold for protein translation. There is some evidence, very few evidence really, that say that circular RNA can be translated into short peptides, and they also can act into the nucleus of the cell by regulating the RNA transcription, interacting with several transcription factors, RNA polymerase II, and so on. So this is the background about the ceRNA network, the experimental model in which we decided to apply this knowledge is meningioma. Why meningioma? Meningioma really is a tumor originating from arachnoid cap cells, forming one of the layers of the protective meninges together with pia mater and dura mater, they represent the most common extra-axial tumor within the central nervous system. So it is an important tumor in terms of rate of incidence. Luckily, most of them are benign, as we'll see in the next slides. Meningioma are often, from a pathological point of view, related to several syndromes, like, for example, neurofibromatosis, Li-Fraumeni syndromes. They can locate in several anatomical sites within the cranium, and rarely they can appear also in extracranial location, in particular in the spinal cord. Not only meningioma represent the most significant in terms of numbers tumor of the central nervous system, meningioma in the last years have an increasing incidence. These are the last statistic from CBitrus that highlight an increase in meningioma incidence of more than 2% in the last 5 years. So the understanding of the causes and also of the molecular mechanisms involved in meningioma biology is very important. Also, these slides show you as meningioma, non-malignant meningioma in particular, represent a big part of this pie chart, where all the central nervous system tumors are shown, are represented. And as you can see here, again, meningiomas are the histopathological tumor more represented among the central nervous system tumors. To connect with what I said before, meningioma classification changed in the last years, and molecular criteria have been recently introduced, most specifically in the last report from the World Health Organization. We can see that mainly grade 3 meningioma have new molecular properties highlighted. For example, molecular mutation of the TER promoter, homozygous deletion of one gene involved in the cell cycle control. So the introduction of molecular criteria clearly say that the better understand deepening the knowledge of molecular aspects of meningioma is critical. Also is critical for all the tumors, but meningioma more specifically, because there was no a molecular classification of this tumor before the last update of WHO classification. On the right of the screen, you can also see other aspects of the molecular biology and the genetics of this tumor, like, for example, the neurofibromatosis 2 gene mutation, and also other genes whose mutation is mainly involved in the so-called NF2 wild-type meningiomas. Why is important to better understand this molecular classification of meningioma? For several aspects. From our point of view, it is very important to better correlate these molecular features with the capability of meningioma to recur. And this is one of the main aspects of this webinar, but also to classify meningiomas in terms of prognosis and in terms of possible therapeutical targets. So, the involvement of several non-coding RNAs, and specifically microRNAs and long non-coding RNAs in meningioma biology, response to therapy, and also relapse is quite known. Several microRNAs have been described as over or under expressed in meningioma tissues with respect to physiological meninges, and the same for long non-coding RNAs. But I think that the most interesting aspect of the molecular classification of meningioma is the integration of omics data. As Bailey and colleagues showed in their article published on Science Advances in 2022, the meningioma can be subtyped in at least three different classes based on specific molecular features in terms of methylation, transcriptomics, cytogenetics feature, and NF2 mutation. From MENG-A to MENG-C, there is like a gradient of malignancy. MENG-A are the most benign meningioma. MENG-C meningioma are the worst in terms of prognosis, and they most probably recur after resection. Independent, and this is another aspect, an interesting aspect of meningioma biology, independent from their U grade and from Simpson grade, the so-called Simpson grade, that is a type of classification that up to date is used to classify meningioma after, in terms of resection after surgery. So the search for biomarkers that can predict the behavior of meningioma and their recurrence in this omics era is for sure one of the gold standard to and recently the group of Rayleigh published an article in which, through a targeted approach, they identified a biomarker based on 34 transcripts that can predict, based on the level of expression of these transcripts the malignancy of meningioma and their capability to recur. This is very important for the prediction also of radiotherapy responses, because some patient that is affected from a grade 3, that is one of the most aggressive meningioma, probably based on the meningioma biology, will not recur after surgery. So this patient could avoid radiotherapy, while, for example, a grade 1 meningioma that is treated as benign could relapse after surgery. And so in that case, radiotherapy should be performed for that patient. But the real big black hole in this scenario is made by the C networks mediated by circular RNAs. So the main aim of our study was to deepen this aspect of meningioma biology, so build this ceRNA network based on circular RNAs, starting from the 34 transcript biomarkers that for henceforth I will name 34HR high recurrence meningioma in terms of microRNAs and circular RNAs that can regulate these transcripts. I will shortly describe the methodology that we used. It is mainly an in silico prediction, so we started from the identification of microRNAs that can target these 34HR MNG transcript. We used several databases, MIRTAR-base that stores information on validated microRNA targets. MIRGene, the database that stores information on the microRNA family, on the belonging of a specific microRNA to specific families. MIRPAT, that is a tool that can perform enrichment analysis in terms of biological processes and pathways in which candidate microRNAs are involved. So, firstly, we create a little database of microRNAs that can impact, that can regulate the expression of these 34 HR MNG. Then we queried other databases, ENCORI database and circular RNA interactome, to predict which circular RNA can sponge the candidate microRNAs previously identified, and so to start to predict the ceRNA network involved in meningioma recurrence, or at least that may control the expression of these 34 HR MNG transcripts. Then we queried Gene Expression Omnibus database to search for dataset that could store transcriptomic data from which identify the most dysregulated genes and the most dysregulated host genes, most specifically of host genes of circular RNAs, and in which also clinical and molecular information could be stored. More specifically, we focused our attention on this dataset identified by this ID that stores data of RNA sequencing from 109 bulk meningiomas. 82% of these are U grade 1, 18% are U grade 2, so from low to medium graded meningioma. And this is perfectly in line with the statistics of meningioma distribution grading in the real world, and about 17% of these samples recurred, while 83% did not recur. Also, these data are in line with the statistics of meningioma in the real world. The average age of patients was 59 years old. In this dataset, molecular information about the Ki67 index, NF2 mutational status and expression, mutational status of non-NF2 genes in NF2 wild-type meningioma, and information about necrosis were annotated in this dataset. So, from our point of view, this dataset was a very interesting dataset from which several information starting from transcriptomic data could be reached. Then we focused on the third part of the methods, that is the experimental validation of the ceRNA network identified through the previous steps. From this point of view, we used the real-time PCR to determine the gene expression level, the expression level of both candidate circular RNAs and the matching host genes. So all this information allowed us to build the ceRNA network that I told before. As Mike told at the beginning of the webinar, I kindly ask you to do not share these data because they are unpublished yet. So thank you for this. About the results. The results showed that our first query, our first search of microRNAs involved in the control of the 34 HR-MNG transcripts gave as an output 32 microRNAs that henceforth are defined as MR. Sorry, not MR, HR, MNG microRNAs or Meningioma recurrence, okay, microRNAs, have been identified as potentially regulating the transcript of the biomarker told before. Among them, the most represented family was that of MIR15, and microRNAs 124 3P was that with the with the highest number of potential targets among the 34 HR transcripts from which we started this research. This is the continue of the table with the other microRNAs identified, and then to first understand if the way that we tried to follow was right, we determined, we analyzed the enrichment of pathways and biological processes in which the candidate MR microRNAs are involved. And the microRNA pathway analysis showed that the microRNAs identified are mainly involved, are enriched in the control of pathways regulating cancer in general and cell cycle most specifically. So the pathway analysis told us that we identified microRNAs involved in pathways strictly related to cancer, and most probably, as they are involved in cell cycle, in cancer relapse. At the same time, the biological processes analysis allowed to identify cell cycle as one of the most regulated biological process, and also cell division. Here we show the top 10 pathways and biological processes in terms of probability of enrichment. Then we focus our analysis on circRNAs. And what we found? We found that 96 circRNAs, as you can see here, are listed in this circle, were predicted to sponge the 32 microRNAs, candidate microRNAs I spoke before. And these 96 circRNAs were biogenerated from 11 of the 34 HRMNG transcripts. 7 of these host genes biogenerated 51 MR circRNAs, showing microRNA binding sites, a number of microRNA binding sites greater than or equal to a median value of 5. So we calculated the median value of microRNA binding sites within the starting 96 circRNAs, and we found that 51 of them had more or equal than 5 binding sites, where 5 represent the median value of binding sites within circRNAs. And what information we obtained? We obtained an information that, as expected, there is a direct correlation between the size of the MR circRNA and the number of microRNA binding sites. The bigger the size of MR circRNA, the bigger is the number, the higher is the number of NDS. At that point, we decided to focus on specific ceRNA network, because data we obtained before gave us a general view of the ceRNA network potentially involved in the control of transcripts that are dysregulated in recurrent meningioma. But what about the feed-forward loop? I mean, what about those ceRNA network that foresees the presence of circular RNAs biogenerated from host gene that are that can sponge microRNAs that in turn control their host genes. From this point of view, we focused our attention on two transcripts. We started from the 34 HRMNG transcripts, and we then focus on two of that transcripts, CHEK1 and PIN1. Both transcripts are involved in the cell cycle regulation, and both transcripts can biogenerate circular RNAs that sponges, that in turn sponge microRNAs regulating the same host gene. ENCORI queuing of ENCORI database allowed to select specific circular RNA isoforms in which we identified specific microRNA binding sites. And microRNAs 16-5P, 124-3P, 193B3P, and 195-5P were the 4 microRNAs potentially involved in these circuitries. Interestingly, all these 4 microRNAs are known tumor suppressors in meningioma, and both CHEK1 and PIN1 are known to be upregulated in meningioma. We decided to filter more our data and to query the dataset of the transcriptomic dataset that I told at the beginning of the methodology presentation. And what we saw was that between PIN1 and CHEK1, the first, PIN1, was the transcript most interesting from our point of view, because it was upregulated, significantly upregulated in U grade 2 meningioma with respect to U grade 1 meningioma, and it continued to raise its level in recurrent meningioma with respect to not recurrent meningioma. The same transcript did not vary its expression in the comparison between recurrent and not recurrent U grade 1 meningioma. So this first query gave us an information about the potential involvement of this transcript in the recurrence of grade 2 meningiomas. As I told before, Bailey and colleagues subtyped meningioma into several subtypes based on several molecular features, and most specifically, what we saw was that in the same dataset in which we analyzed PIN1 gene expression, the MENG-C meningioma, the most aggressive phenotype, if you remember, is the most represented in recurrent meningiomas, while the MENG-A meningioma was the most represented in the not recurrent meningioma, as expected, and quite same distribution. As you can see, there is a higher proportion of MENG-C meningioma appears in U grade 2 recurrent meningioma with respect to grade 2 not recurrent meningioma. Why I show you this slide? Because as you can see, the PIN1 expression follows this scheme, follows this subtypization. The more aggressive the meningioma subtype, based on Bailey classification, the higher is PIN1 expression. And you can see that MENG-C meningioma subtypes shows the higher level of expression with respect to MENG-B and more specifically to MENG-A in terms of statistics. We decided to deepen this aspect, and before build the ceRNA network, we decided to see if PIN1 was really related to a most aggressive and most probable recurrent meningioma. And we decided to investigate the relation with other molecular feature, like, for example, chromosome 1p loss, that is, and also chromosome 22q loss, that are two features of the more aggressive meningioma. As you can see here, there is general increase in the level of PIN1 in chromosome 1p loss and chromosome also in chromosome 22q loss meningioma with an higher fold change difference in grade 2 meningioma. Grade 2 meningiomas show the highest level of PIN1 expression with respect to their matched controls. In this case, controls are obviously meningiomas that do not show chromosome 1p or 22q loss. Another phenotypic feature that we investigated was necrosis. Also, in this case, as you can see, grade 2 meningioma shows significantly higher level of PIN1 expression in necrotic as compared to not necrotic meningioma. I remember that necrosis is another feature typical of the worst phenotype of meningioma. And also, by correlating the PIN1 expression with NF2 and Ki67 expression, we saw that in grade 2 meningioma, more specifically, there is a significant negative correlation between PIN1 expression and NF2 expression. I remember that the lower the expression on NF2, the worse is the malignance, the phenotype of meningioma, and there is instead a borderline significant positive correlation between one expression and Ki67 expression. Ki67 is an index, represents an index of proliferation of the cells. So the higher is the P1 expression, the more meningioma cell proliferate. So based on these data, we decided to deepen the knowledge of molecular network potentially regulating PIN1. And so we went back to the first question. Is there the possibility that through a feed-forward gene expression loop, PIN1 expression may be controlled in recurring meningioma? Going back to the previous slide, I remember that two microRNAs, microRNAs 16-5P and 124-3P, were predicted to regulate PIN1, and at the same time, they may be sponged by circPIN1 circular RNAs. And also the same for microRNA 195-5P. To build a reliable ceRNA network, it would be important also to check not only the predicted binding of microRNAs to the sponging non-coding RNA molecules, but also to see correlation between the non-coding RNA regulator, in this case the circular RNA, and the mRNA involved in the circuitry, in this case PIN1. So we decided to check for correlation between the expression of circPIN1 circular RNAs, and more specifically, we identified the two isoforms shown in this graph, because they were the only two isoforms expressed in the tissues that we investigated. And as you can see, there is a strong positive correlation between the expression of the two candidate circular RNAs, and positive correlation is also present between each of the two circular RNAs. Here it is shown the first one with PIN1, and here is shown the second one with PIN1. There is a strong positive correlation also because circular RNA and PIN1 expression. Furthermore, there is another aspect that we consider crucial, critical for the identification of a reliable ceRNA network, that is the capability of agoproteins to bind the circular RNA. Because I remember that when the microRNA recognize its target or its sponge, this recognition is mediated by agoproteins. And what we saw was that agoproteins, AGO1, AGO2, together with other interesting RBP proteins that we are also investigating in this project, are statistically significant, more represented in terms of RNA binding protein binding sites with respect to other RNA binding proteins investigated starting from a total of 140 circular RNAs with a same size of our two candidate circular RNAs. And furthermore, we also investigated the expression of microRNAs, the two candidate microRNAs, into tissues strictly connected with meninges. And as you can see, both microRNAs, although at different level, are quite highly expressed in all these meningeal tissues. And also PIN1, whose expression was investigated at single cell level, and more specifically in fibroblasts of proliferating meninges, showed that PIN1 was expressed in proliferating meninges, and this is indicative of its potential involvement in meningioma. In conclusion, these studies show how PIN1 could be considered a new candidate gene involved in meningioma recurrence. PIN1 is also predicted to be regulated through a feed-forward gene expression loop made of the two circ1 circular RNAs, MIR16 5P, MIR19 5P, and also this study paves the way to deepen a new chapter of meningioma biology in terms of circular RNA-mediated ceRNA networks. Obviously, this study shows several limitations. First of all, it is based only on one dataset. I mean, this is due to the difficulty in retrieving a complete dataset publicly available on that matches transcriptomic data with all the 3 grades meningioma and also with clinical and molecular data, and lack also of experimental data in meningioma biopsies and mechanistic studies of in vitro cell models. We are performing these last experiments, and this is the point. The last slide, probably the most important slide, is about the acknowledgements. I really say thanks to Dr. Giuseppe Sotera, that is a medical doctor that is involved in this study, and we started with him this study, and he continues to spend lots of his time and efforts in this study. All the other members of the lab, and obviously the University of Catania, and my department for hosting me, and the department of other department involved in the collaboration, the department Gianfilippo Ingrassia, where Professor Barbagallo and Professor Caltabiano and Professor Brogi are working and are sharing with us their samples and expertise, and the funding, thanks to Piaceri funding, that is an internal university grant that allowed us to perform the experiments, some of the experiments that I show here. Thank you, all of you, for your patience in for this presentation.
Thank you very much, Davide, for a fantastic talk. I really enjoyed it, and I'm sure the audience did as well. Really, really fantastic, interesting science. I'm informed that we do have time for some questions and answers, and we might go beyond the hour. I hope that's okay with you, Davide. If anyone needs to leave, then obviously we won't be offended. But if anyone is happy to stay for a little bit longer for some questions, that would be fantastic. So I can see 3 questions in the Q&A. So if it's okay, I will read them out too. So first is from Muhammad Salman, who asks, what happens if a meningioma is not removed? If I maybe try to interpret that, if I may, Muhammad, maybe the question might be around, are there differences if there isn't surgery or if there isn't radiotherapy or kind of intervention? What happens to these ceRNA circuits?
Yes, it's an interesting question. It's difficult to predict because obviously, if we do not remove the tumor, we cannot analyze it apart from biopsies, eventual biopsies that can be taken from the patients. But I think that we can answer to this question only if we experimentally validate this. There is an aspect, an interesting aspect related to this question that is about imaging, because probably imaging can solve this problem, because thanks to investigation of magnetic resonance imaging, you could predict the behavior of this tumor without surgery. So probably the answer is this.
Thank you. So we have a question from an anonymous questioner, which is circular RNA and technically a non-coding RNA? Maybe if I can also interpret this, if I may. What is a circular RNA essentially, and is it strictly non-coding? Is there any evidence for open reading frames? What do we know about their nature?
Yes. As I told, thank you for the question. As I told during the presentation, there is very few articles speak about the possibility of circular RNAs to be translated. Some of circular RNA show open reading frames that can be translated into proteins, probably through an internal ribosome entry site mechanism, but there are no more information about this.
Thank
so
you.
They essentially could be categorized as mainly as non-coding RNAs.
Okay, but maybe there's some functional versatility there. But yeah, okay. Thank you. The next question is from Adiba Shakil. So it's in two parts. Can ceRNAs be secreted or associated with exosomes? And also, does the PIM1 cirRNA network affect the surrounding microenvironment or immune response in the tumor?
Oh, this is a very interesting question. Thank you. About the first one, circular RNAs can surely be secreted by exosomes. We published some papers about this aspect, and the pathway through which this happens is unknown yet, but they can be found into exosomes and they can be secreted. About the second question, sure, PIM1 can affect the microenvironment, most probably, because as I show in one of the last slide, it is quite highly expressed in fibroblasts associated tumors, in cancer-associated fibroblasts in CAFs. So probably it interacts with the tumor microenvironment, and this is an interesting aspect that we will deepen in the next future.
Thank you. We've got a couple more questions. So a question from Siti Hamidatul Aliyah. What are the differences in microRNA expression between tissue and circulating samples in the same patient? And how might these differences impact the use of microRNAs as biomarkers for diagnosis and monitoring? Is there a correspondence between the tissue or tumor and circulating material?
Circulating. Okay.
Yeah.
It depends. There is not a rule from this point of view. Based on my experience and also based on literature, some microRNAs that is upregulated in the tumor tissue could be found as down expressed in exosomes or as a circulating molecule. It probably depends on the some yet unidentified molecular aspect that can retain, for example, into the cell the overexpressed microRNAs, not allowing them to escape the cell and to be released in extracellular fluids. Probably these are the mechanisms. So there is not a strict correlation.
Thank you. One or two more questions. Question from Youssef Sadallah. If I understand this correctly, when we talk about circular RNAs, do they can they have epigenetic functions?
Oh, sure. It depends on what you mean about epigenetics, because if you mean epigenetics in terms of, for example, the possibility to change the status of methylation of DNA, probably through indirect mechanism they can act. But surely, for example, I show the slide of one of our paper published on the International Journal of Molecular Sciences, where we identify the microRNAs that circular RNA, sorry, that can bind a splicing factor, and so it can epigenetically control the expression of several transcripts. So yes, they can for sure be involved in the epigenetic control.
Thank you. The next question is a cracker. It's a great question. How can physicists, and this is by Gaurav Kumar Gupta, how can physicists and mathematicians contribute to simulating ceRNA networks or predicting cancer-related outcomes based on transcriptomics data? What is the impact and challenge of AI being faced or applying to the field? Sorry, I don't know if I expressed that well. Yeah.
I think that mathematics and physicists play, and AI for sure play a critical role and will play a critical role in better refine these mechanisms, because I told very easily about the ceRNA network, but they are very complex really in terms of correlation of gene expression at tissue level and single cell level. So the more data we will acquire in terms of single cell RNA sequencing, I think the more we can predict a reliable ceRNA network. And for sure also building a mathematical model for this network will be a critical point.
Thank you. If I may indulge and ask a question or two myself quickly. Davide, at what point does stoichiometry become important? I mean, because we have this incredibly complex soup of regulatory RNA networks, right? But surely the relative levels are going to be important in terms of the likelihood of interactions and so on. So how crucial is stoichiometry here?
For sure, stoichiometry is crucial. I think we don't perform absolute quantification. So I can't say how many molecules of each of the actors is present into a cell and which is the cutoff of this level. But I think that we can answer to this question through the functional experiments. So if you alter the expression through in vitro models of one of the actor, what is the effect on the network you are investigating? So probably the stoichiometry is for sure important, but to find the cutoff, I think it is very difficult up today. Probably the most effective way to say if the ceRNA network you found is reliable is to perform this type of experiments that artificially alter the expression of one of the actor to see what happens in the entire circuitry.
Thank you. We have one more question from Rashmi Rana. Does the overexpression of ceRNA connected to angiogenin-mediated tumor progression?
In this case, in general, probably yes, because it depends on the target of the cRNA network, obviously. In glioblastoma, for example, we show that the circular circRNA-5 that you cited at the beginning of the webinar is involved in the regulation of the splicing of the vascular endothelial growth factor, and so it is connected, it indirectly controls the angiogenesis, but obviously it depends on the target of specific circular RNA. In the case of meningioma, we have some data that I didn't show that say us that probably these two circular RNAs are also involved in the control of angiogenesis, but it depends obviously.
Thank you. Perhaps I'll just ask the very final, quick, obvious question, which is, how does this ideally develop for patients in 10, 20 years? You know, how does this translate to tangible benefit for patients?
First of all, I think in terms of therapy, because identify a central hub in a network is crucial to find a reliable therapy, I think, for the tumor cells. So, in terms of therapy, and by deepening the study in terms also of sequencing, for example, I think in terms of personalized medicine, and so also prognosis of the could be another goal that would be achieved in the next years.
Thank you. Thank you very much. We will have to conclude the webinar here because otherwise they'll get in trouble. We are 10 minutes over. So I'd just like to end by thanking everyone involved. First and foremost, Davide, thank you very much for a fantastic talk. Thank you, Daniel, for and the ncRNA staff for organizing the event, and the wider MDPI staff. Thank you very much for facilitating this. And thanks to all of you for attending, for the great questions. I hope you enjoyed the webinar, and hopefully there will be others and you can join us again. Thank you very much, everyone, and have a wonderful evening or morning or wherever you are in the world. Thank you very much.
Thank you very much, Mike. Thank you, Daniel. See you. Bye-bye.
Bye-bye.
Thank you, everyone. Thank you.